this is basically the formation of glycogen, right? So you see here, this is one alpha glucose molecule and this is second alpha glucose molecule. So this is carbon 1, right? 2, 3, 4, and 5, and then 6. So the carbon 1 of the first alpha glucose is actually combining with the carbon 4 of the second alpha glucose molecule, right? Did you get it? Now these two are forming the glycosidic bond, 1,4 glycosidic bond, right? So this is the 1,4 glycosidic bond formed between these two molecules, right? Now what is happening here? This 1,2 glycosidic bond is also forming a branch here, fine? So this is actually the 6th carbon, fine, and this is the first carbon, right? So now if this bond is formed, this OH will be spare, fine. Now this OH is going to form bond with another OH of this group, right? With the 6th group, fine. So you see here, this one is free actually, fine, and this one is free. So this is a disaccharide. Now this disaccharide is again going to branch with this first carbon and maybe the 5th, 6th carbon of the next molecule. So there's going to be a branch formed like this. You see here, these are alpha 1,4 glycosidic bonds in this branch, fine. And then this 6th carbon of every alpha glucose molecule is binding with another chain, with the first carbon of another chain. So these are two types of bonds in glycogen formation: 1,6 glycosidic bonds and 1,4 glycosidic bonds. 1,4 are in the straight line chain and 1,6 are in the branches. So whenever the glycogen molecule is forming a branch here, it is forming the alpha 1,6 glycosidic bond. Okay?
Yeah.
You see two different molecules here. Now here, these are two alpha glucose molecules combining through two different glycosidic bonds, 1,4 and 1,6. Now in case of cellulose, this is opposite. You have two beta glucose molecules, but this beta and this beta cannot combine because you see here, this OH is at this position and this OH is at opposite position, right? Do you get it?
Yeah.
Now what will happen here is that this OH is going to invert. So this will be inverted beta glucose. Now this OH comes here, fine. So you see here, this is inverted beta glucose. Okay. Now these two inverted beta glucose, these OH molecules will combine together, fine, and they'll form glycosidic bond, which would be called 1,4 glycosidic bond, because this is between 1 and 4 carbon right now. But in this case, you have one beta glucose and one beta inverted glucose, right? So this chain is now called cellulose. So this is a big difference between glycogen and cellulose. You have 1,4 glycosidic bond between alpha carbons here, and you have 1,4 glycosidic bond between beta glucose and beta inverted glucose here. And another major difference is that you don't see any branching here, but in case of glycogen, you have branching because of this alpha 1,6 glycosidic bond. Okay.
Okay.
So you have to keep this in mind. Okay. They can ask you for differences between the glycogen and cellulose as well. So you have to imagine that when glycogen is formed, it is actually a highly branched structure, okay? So these branches are formed because of this 1,6 bond. 1,4 bond will be in every carbohydrate polysaccharide chain, okay? Whatever the carbohydrate is, if it is forming a disaccharide or a polysaccharide, you will see this 1,4 glycosidic bond. But you will see this 1,6 glycosidic bond in few cases only. For example, you see them in glycogen. You might see them in amylose and amylopectin, sorry. Fine? You see here? Amylose here, fine. So this amylose and amylopectin, those are also branch structures. So they will also be having this 1,6 carbon bonding. So whenever a polysaccharide wants to form a branch, the branch will be formed because of this 1,6 glycosidic bond.